Hey, this is Mrs. Reichelt, and our next video we're going to talk about is the humoral immune system. So last video we talked about there's two different types of the adaptive immune system. You have humoral and cell-mediated. So this one is targeting the humoral, or we're attacking pathogens only, this one. So let's go ahead and get started. So the B lymphocytes have specific receptors that bind to a specific antigen. We sort of talked about this last video because I said if this was the cell, or if that was the pathogen, maybe a virus or something, it is going to have a special shape. And the B lymphocytes are going to kind of adapt to that shape so they can specifically target that pathogen. The binding event will activate the lymphocytes to undergo a clonal selection, meaning that it's going to make a little clone of part of the surface antigen so it can clone them and then attack them later. A large number of the clones are produced in the primary humoral response. All right, so most B cells become plasma cells. They produce antibodies that destroy antigen, and their activity lasts between 4 and 5 days. And then some B cells are going to become long-lived memory cells. Remember last video we talked about how some B cells have memory? So some of them are just going to be used in the antigen, or I guess the antibody response, and then some of them are going to be converted into these long-lived memory cells. We call that the secondary humoral response. So the secondary humoral response, you have memory cells. A second exposure will cause the rapid response of those initial antibodies, and then the secondary response is stronger and it's longer lasting. So basically what that means is, I'm going to show you a little picture here. So basically if we have our antigen in this diagram are going to be these little orange circles. Okay, so an antigen will bind to a receptor, just like this, okay, and on a specific B cell. What happens then is those B cells are going to make several clones. This little thing is the actual antibody, that it's going to have this special shape so that the antigen will bind onto that so they can destroy it. Excuse me. So basically, once you have that antigen, you have a B cell that's going to make a special shape of an antibody that will specifically bind to that antigen. From there, you're going to make clones of that B cell. The purpose of making the clones is so that it can attack all of those antigens. Some of those B cells are going to attack the antigens, and you can see the antigen being assembled right over here. But some of them are going to be converted into memory B cells. So the purpose of them being converted to memory B cells is that if you're exposed to that same virus or that same type of infection again, your body will remember what that was like. And I'll show you what it looks like right here. So this is your relative antibody concentration in blood plasma. So when you have, let's say this is when you're injected with an antigen. So you are injected with a virus, we'll say. So you have a virus, and after 2 weeks, your primary response of the concentration of antigens is about here, and there's no number associated with it. But then let's say you get better and your virus kind of goes to bay, and then all of a sudden a couple of weeks later your body is responding to that same antigen. Well, now all of a sudden, because of your memory B cells, your antigen concentration goes from 0 all the way up to here. And I know this isn't the best graph because it doesn't show you what the actual concentrations are, but hopefully you can see the difference between primary responses here, secondary responses all the way up here. So basically the big takeaways I think for the humoral response is to remember that the humoral response is going to attack antigens or pathogens. That is a big deal. Okay, it's going to occur in the body fluids or in the humor. That's different from cell-mediated immunity, because cell-mediated immunity is going to attack infected cells. So cells that are already infected by the pathogen, it attacks those. So that's the major takeaway, and I hope you found that helpful.